Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss examples of programmed cell death events. When there is extreme physical, chemical, or mechanical damage to a cell, then the cell dies by a process that we call accidental cell death or ACD. This type of cell death is unregulated and the cell typically dies by a process called necrosis. Besides accidental cell death, cells can also die by programmed cell death or PCD. Unlike ACD, this is a regulated process. It's under genetic control. And in this case, the cell can die by a process called apoptosis or several other types of PCD that we'll discuss in the next video. Let us look at examples of programmed cell death. Let's first look at a few examples of programmed cell death that happens during development. Programmed cell death or PCD is important for morphogenesis. For example, excess cells between the fingers and the toes. These are removed by PCD so that we can generate these structures that we call fingers or toes. During development, excess neurons that do not form successful connections with target cells, these neurons will die by PCD. PCD plays a very important role in the generation of a healthy immune system during development. For example, between birth and puberty, the thymus plays an important role in the removal of autoreactive T lymphocytes that will attack our own cells. The immature or the developing T cells, what we call thymocytes, these cells will interact with surface proteins present on the thymus epithelial cells. These proteins are MHC self-antigen complexes that are present on the thymus epithelial cells. In the cortex of the thymus, those developing T cells with correctly formed T cell receptors that recognize host MHC proteins will survive, while those that do not recognize host MHC proteins, they will die by PCD, especially by apoptosis, and this is called positive selection or MHC restriction. In the medulla of the thymus, the developing T cells that react with self-antigens, they will die by PCD. And this is called negative selection or deletion of autoreactive T cells. So the outcome of this education of T cells in the thymus is that only T cells that would not attack host cells or host antigens, that means self, but only will attack foreign cells or antigens, non-self. Only these cells will survive. The rest of the T cells which do not do this, they will die. And this is important because we don't want our immune system to attack our own cells. We want our immune system to only attack foreign cells. PCD also plays an important role in normal physiology. For example, in the menstrual cycle, when the hormones estrogen and progesterone levels start decreasing, the hormone-sensitive cells in the endometrial functional layer, the epithelial cells as well as the stromal cells. These cells will undergo programmed cell death, and this will lead to the breakdown of the lining of the uterus in the menstrual cycle. Another importance of PCD is maintenance of organ size and shape. Organs reach their correct size and shape during development and throughout life through a balance of cell proliferation as well as programmed cell death. PCD also plays an important role in the elimination of excess activated effector T cells. So, for example, during an infection, we have microbes. Of course, they are releasing all kinds of antigens which are present on their surface or being secreted. A lot of cytokines are being secreted by various immune cells. And all of this will lead to the proliferation as well as activation of effector T cells, which are basically lymphocytes. Now, once the infection is resolved, the infection is cleared, there is no more microbes present, the cytokine production goes down, and now these T cells proliferation will start decreasing. So after the infection is resolved, we still have all of these activated effector T cells hanging around. And that can be dangerous because these T cells can attack our own tissues. They can cause collateral damage to our healthy tissues. So it's important that in order to prevent an overactive immune response, these T cells need to be killed off. So in order to kill these cells, what happens is we have these regulatory T cells, and these T cells have a signal, a ligand called FASL. And we'll talk about this under apoptosis in a future video. And these cells will bind these activated effector T cells, and these effector T cells have a lot of these receptors here called the FAS receptor, the FAS receptor. So a lot of highly expressed in these activated T cells. So when the regulatory T cell binds the effector T cell, these two will interact, and that will trigger programmed cell death. It will trigger apoptosis, something we'll talk about in a future video. So this way the excess effector T cells are killed off to prevent an overactive immune response from causing damage to our healthy tissues. A few examples of the importance of PCD in pathology when our cells are stressed. Stress cells are killed off by programmed cell death, and that's important to prevent accumulation of mutated cells or infected cells or damaged cells. Now, there are several different factors that can cause a cell to be stressed. So, for example, when there is growth factor stimulation or lack of stimulation, that can lead to stress. Most often, growth factors cause cells to survive. However, there are cases where growth factors can actually cause cells to undergo programmed cell death. More commonly, lack of stimulation by growth factors is the reason why cells can undergo programmed cell death. Infected cells or abnormal cells, such as cancer cells and damaged cells, are also stress cells. reactive oxygen species, oxidative stress that we talked about in a previous video, which can be due to many different factors, including iron excess. This is a common cause of cell stress. Another cause of cell stress is energy deprivation, for example, lack of oxygen or lack of the fuel glucose. As we shall see in a future video, copper excess is also a cause of cell stress, which can lead to programmed cell death. DNA damage as well as defects in DNA synthesis or replication or in cell division, these are all causes of cell stress. ER stress is another cause of cell stress. In this case, the endoplasmic reticulum is overloaded with proteins that are unfolded or misfolded, and this can lead to programmed cell death. Another cause of cell stress is increase in intracellular pH, what we call as intracellular alkali excess, and this can also lead to a type of programmed cell death that we'll discuss in a future video. And then finally, we also have medical treatment such as the use of drugs as well as radiation therapy that can also cause a cell to be stressed. So PCD is important to eliminate infected cells. So if a cell that is infected, if it cannot fight off the infection, it will die by PCD. And in the process, it will release a lot of pro-inflammatory cytokines as well as chemicals, what we call as damage-associated molecular patterns, or DAMPs. And examples of DAMPs is DNA, interleukins, various interleukins, as well as other proteins, ATP, even some carbohydrates. And these DAMPs will, that are released by the dying cell, they will attract the other immune cells in order to prevent the infection spreading to the other cells. Cells that have problems with their cell division, they will also die by PCD. We saw this in video 8.1. For example, if there is DNA damage, this damage is sensed by this protein mRN, and once the mRN detects the DNA damage, the mRN will bind and activate another protein called ATM. ATM will then phosphorylate and activate another protein called CHK2, and then CHK2 will phosphorylate and activate another protein called P53. And this P53 will now trigger programmed cell death such as apoptosis. So any cell that has problems with cell division and it cannot resolve that problem, that cell will eventually die by programmed cell death. Another example of programmed cell death is what we call as the unfolded protein response, or UPR. Recall that when the polypeptide chain undergoes synthesis, it must also undergo folding, proper folding, to give the correct three-dimensional shape so that the protein can carry out its function. Now, several factors such as nutritional problems, hypoxia, mutated proteins, impaired chaperone activity. Recall that chaperones are proteins that help other proteins fold correctly. Pathological triggers, all of these factors can lead to abnormal protein folding, and that can lead to the accumulation of unfolded or misfolded proteins. And when these proteins accumulate in the endoplasmic reticulum, where a lot of protein synthesis happens, then that leads to what we call as endoplasmic reticulum stress, ER stress. So a cell under ER stress is going to try to stop the accumulation of these abnormal proteins in the ER lumen. These misfolded proteins are sensed by ER chaperone called BIP-GRP78, and this will trigger cytoprotective signaling pathways. And these pathways will restore protein folding homeostasis, what we call the unfolded protein response. So the UPR hopefully will reduce further protein synthesis and will destroy any misfolded proteins present in the ER or elsewhere in the cell. There are several proteins that participate in the UPR, and examples are given here. So, for example, this protein PRK, this will inhibit the translation of initiation factor, and without initiation factor, further protein synthesis is prevented. Another protein called ATF4, what it does, it increases the antioxidant capacity of the cell because a lot of this misfolded protein problem is caused by oxidative stress. So by increasing the antioxidant capacity, it can reduce the oxidative stress and thereby reduce further protein misfolding. ATF4 also initiates macroautophagy, and this autophagy will eat up or destroy the misfolded proteins. Another protein that plays an important role in the unfolded protein response is the IRE1, which is inositol requiring protein 1. And IRE1 basically what it does is that it will help take all the unfolded proteins in the ER and send it to the cytoplasm. And in the cytoplasm, these unfolded proteins will undergo degradation by the proteasome. The IRE1 will also stop further proteins from entering into ER. And then the protein ATF6 will help IRE1 to carry out its function. So hopefully with all of these activities, protein synthesis will stop, and all the misfolded proteins present in the ER will be either broken down or sent to the cytoplasm and broken down by the proteasome. And hopefully this will restore the protein folding homeostasis and bring back the cell to its normal state. However, if the ER stress persists, then these proteins PRKRE1 will will increase the transcription factor CHOPGADD153, and this transcription factor will increase proteins that will cause apoptosis, will cause the cell to die by apoptosis. So a cell under ER stress will try to overcome its stress by stopping protein synthesis and getting rid of the misfolded proteins. But if that does not succeed, then the cell will undergo programmed cell death, apoptosis. As a side note, it's important to appreciate that increased intracellular accumulation of abnormally folded proteins is commonly seen in several neurodegenerative diseases such as Alzheimer's, Huntington, as well as Parkinson's. And finally, the last example we'll discuss is a disease called autoimmune lymphoproliferative syndrome, or ALPS. This is a very rare pediatric disorder, about 1 in 100,000 cases. This disease involves mutations in certain genes that does not allow programmed cell death, especially apoptosis, to occur in activated lymphocytes. As we discussed earlier, after an infection is resolved, the activated effector T cells are eliminated by apoptosis in order to prevent an overactive immune response and collateral damage that can be caused by these T cells to healthy tissues. So in ALPS, there is a mutation in the FAS receptor. Okay, so 75% of these cases, there is a problem with this FAS receptor. This is a receptor that is highly expressed in the activated T lymphocytes. So when these receptors do not work, these lymphocytes, effector lymphocytes, do not undergo apoptosis. Okay, so after the infection is resolved, these cells do not die. And without apoptosis, these effector T lymphocytes will start accumulating, especially in lymph nodes and spleen. The lymph nodes will become very large, what we call lymphadenopathy. The spleen becomes very large, what we call splenomegaly, and that's what we see in these patients with ALPS. The presence of these activated T effector cells may also lead to the attack of the body's own cells, such as red blood cells and platelets, and then these patients might show autoimmune disorders such as hemolytic anemia, as well as immune thrombocytopenia. So in hemolytic anemia, the red blood cell count goes down. In immune thrombocytopenia, the platelet count goes down. So hopefully these examples will help you appreciate the importance of PCD in health and disease. In the next video, we shall discuss the different types of programmed cell death. So until the next video, once again, thank you very much for watching and supporting this channel. Goodbye and have a wonderful day.